Welcome to AHDB Agronomy Week 2020. The live stream session will start shortly. Before we begin, we wanted to remind you of a few points of housekeeping. You're all on mute, so don't worry, we can't hear you. The session is scheduled to last between 1 and 1.5 hours, including questions. We want this session to be as interactive as possible, so please post your questions throughout the session using the live Q&A function in the Agronomy Week platform below. We're recording this session, so if you miss anything or would like to watch it again, it will be available on the AHDB YouTube channel and AHDB website. You can also come back and watch the recordings on the Agronomy Week platform for 3 months. At the end of the session, we'll provide you with unique BASIS and Naroso codes. Don't forget to complete the BASIS and Naroso forms using the relevant tabs on the platform. You have 2 weeks after the live session to register for your points. Join in the conversation online. Follow AHDB_cereals and AHDB_potatoes on Twitter and use the hashtag AgronomyWeek. If you have any issues with the conference platform, there are digital event FAQs in the menu on the left-hand side. You can use the Help tab to contact the team if you experience any technical problems during the week. We hope that you enjoy Agronomy Week 2020. Thanks for joining us. Your session will now start.
Good morning, everybody, and a very warm welcome to Agronomy Week 2020. Delighted that this is the first session of what's promising to be a really exciting week laid out ahead for you. Today and tomorrow is all about cereals and oilseeds, and the rest of the week about potatoes. My name's Teresa Meadows, and I'm the Knowledge Exchange Manager for AHDB Cereals in East Anglia, and it's my pleasure to be hosting this session where we're focusing on growing oilseed rape and the different areas that we can look at improving as agronomists, as farmers, as new agronomists, as BASIS qualified advisors, or for those across the industry and are joining us today. Today's session is looking about oilseed rape, and in particular, how we can achieve our best yield potential and focusing on flea beetle and larvae control. There is a reason for doing that, and as we all know, the landscape of growing oilseed rape has changed a lot in the last few years, particularly for those of us in East Anglia, where cabbage stem flea beetle and the management of that has become much more of a challenge than normal. This landscape, David Woolston's picture there, looking at a companion crop growing with oilseed rape, is just one of the ways that oilseed rape has been growing slightly different, and you as agronomists, as farmers and advisors are looking at how we can manage that. Whether it's leaving stubble longer, whether that's rolling, whether that's managing oilseed rape with your neighbours and looking at flea beetle levels, whether that's managing it within your rotations. Oilseed rape is still really important in the crop, and if we can have a good crop, then it makes a big difference to the bottom line. We are very conscious, however, that this year cabbage stem flea beetle and the adults and the feeding in August and September wasn't as much of an issue, and so we've got some good crops that are there available. There were some that kind of got later in the year, but we have actually got really good and really lush crops available now. And so we thought we'd focus this session on the latest AHDB research and research from across the industry on actually how we can manage the larvae, what we should be looking for in terms of the larvae this year, and managing those lush crops. Richard Ling, our DIS monitor farmer, is there in a crop of his, grown with a companion crop, but actually that crop is possibly going to present different management decisions this year to some of the smaller crops we've had in the past. And so that's why we thought we'd bring in Sarah Kendall from ADAS, that's done a piece of research funded by AHDB, looking at what has led to higher yields in those that are entering the yield enhancement network, the YEN. And so Sarah, along with Sacha and Sam and Charlotte, are going to give us an insight into these topics, hopefully applicable for you for the year that you are facing ahead of us. So the format for today's session is I'm shortly going to hand over to Sacha White. Sacha's going to talk us through the work the AHDB funded and wider work in the industry about integrated pest management actions, looking at larval control, looking at defoliation and trap cropping, areas that he's been looking at. We're then going to move on to Sam Cook, who's joining us from Rothamsted Research this morning, and her focus is going to look at, again, larval infestations in oilseed rape, the work that the research they've been doing, looking at mitigation measures, some of the novel research and what you can do on farm to help with that. Charlotte Rowley is then joining us, who's our AHDB research lead in this area, and is going to talk to you through some of the wider projects that you can look at for further research in this area. We're then going to have a Q&A session focused on what you've heard. And so if you've got any questions, as Emily just said, please put them in the chat that you can see just below where we are this morning. We're then going to move on to Sarah Kendall, and Sarah, as I said, is going to present the lessons from the Oilseed Yield Enhancement Network data and the results that have come from that analysis. And then we'll have a plenary session with all of the speakers to pick up any questions that you've picked up through the session or anything else you might like to ask, finishing this morning at 12:30. So, just interested, and while we're running through Sacha, Sam and Charlotte's presentations, there is a poll. If you'd like to go to the pollev.com/AHDB or click on the link that you can see just below this video, we'd like you to answer whether you've been out and had a look for larval damage in your fields, and if you have, what kind of larvae you're finding in the plants at the moment. It'd be interesting to hear the picture from across the country, and if you've got specific kind of thoughts or things you found, please feel free to put that in the questions box as well, and that will give us a feeling of what we're finding. Equally, if there's any comments about the rate that you've got in the fields or questions, please pop those in the questions box as we go through. So, without further ado, I'm going to hand over to our first speaker and to Sacha White from ADAS. Sacha is a senior research entomologist working with ADAS with over 10 years experience working within the arable and horticultural crops. His focus is around integrated pest management and managed many of our AHDB research projects with a look at cabbage stem flea beetle. So I'm pleased to hand over to you, Sacha, for this first session and your presentation.
Thanks, Teresa, and morning, everybody. So I'm going to be talking about ways that we can use IPM to manage flea beetle larvae. And I should say before I start that almost everything that I'm going to be talking about has come out of this project here. It finished earlier this year. It's the cabbage stem flea beetle IPM project. And that project looked at a whole range of things, not just larval control, and it culminated in this flow diagram of control options that you can see here. So these are control options that you can use at different points throughout the crop life cycle, and each control option was given a traffic light system. So essentially a green light meant that that option provides good control and we've got good evidence for it. Orange means there's indications that it provides good control, but we need a little bit more research. And a red light means there's some suggestion that there's a benefit of this, but we need a lot more work to understand how best, what level of benefit it provides. Now, there's a heck of a lot to take in there. I don't expect you to, but it's all available here at this link. That's where the final report is. Or you could just Google AHDB, cabbage stem flea beetle IPM, and it should be the top search result. So the factor that has the biggest impact on your larval pressure is the date the crop is sown. And that's illustrated with this graph here, which shows the autumn larval populations in crops sown during different periods. What you can see is that crops sown at the beginning of August have the highest larval populations, and the larval populations reduce the later the crops are sown, so that by the second half of September, you're getting roughly 50 times fewer larvae per plant than you would in a crop sown at the beginning of August. This data is survey data, but it's survey data from over 1,100 sites across the country in over 10 years. So it's a pretty reliable data set. The reason we see this effect is because, well, there's two reasons really. Firstly, adult flea beetles are going to migrate into earlier sown crops, earlier than they would be moving into late sown crops. That means that they're going to be laying their eggs earlier. Those eggs are going to be developing in warmer temperatures, which means that they will develop more quickly, they'll have a higher survival rate, and the adults will lay more eggs. The other reason is that crops sown in the second half of September are likely to emerge after the peak of flea beetle migration has passed, which means they're likely to get less adults in them and therefore less eggs and less larvae. We've also seen a trend in recent years for increasing seed rate to help compensate for losses to flea beetle. So in the previous project, we looked at the benefit of increasing seed rate in 4 trials. Each trial looked at 5 seed rates ranging from 10 to 120 seeds per metre squared. In terms of larvae, we saw very little benefit of increasing seed rate, and this is illustrated with this graph on the left here, which shows the larval populations in January 2018 in our trial in Yorkshire. What you can see is the larvae per plant in each of the 5 seed rates. And at the bottom, you can see the seed rates, and under that, in the number in brackets, is the plants per metre squared achieved at that seed rate. And clearly, at this site, what we had is a high level of volunteers in that field. But what we can see is essentially at every seed rate, we're getting roughly 5 larvae per plant. There's no, and that's surprising. What you would expect to see is more larvae where there are fewer plants. That didn't happen, and what this indicates is that flea beetles are making choices. So either the adults are making their choices about where they lay their eggs, maybe not laying eggs around plants that already have a lot of eggs around them or already have a lot of larvae in them, or indeed the larvae are making choices not to crawl into a plant that's already heavily infested. This trend was seen in all of our trials. So essentially we saw that there was no increase in larvae or decrease in larvae with increasing seed rate. Now, whilst increasing seed rate may have no benefit for larval pressure, it may actually have a downside, because as we're seeing at roughly the same number of larvae per plant, regardless of seed rate, but at higher seed rates, you get more plants per unit area. That means you get more larvae per unit area. And that's illustrated with this graph on the right here, which shows the numbers of larvae per metre squared in that trial. What you can see is the numbers of larvae are increasing rapidly as seed rate increases. And this is a trend that we saw again in all of our trials. Now, while this may not affect that crop per se, what it will mean is that that field will have more adults emerging from it in the summer, and therefore following crops might be at higher pest pressure. Another way that we can manage or reduce larval pressure is using trap crops. And we looked at using volunteer oilseed rape as a trap crop. So simply by leaving volunteers that have appeared in fields following an oilseed rape crop, leaving those in the ground undisturbed, what they do is they act as a trap crop. They draw migrating adult flea beetles into that field of volunteers rather than into a nearby field of sown oilseed rape. Where we looked at this in trials, we saw an average of 63% fewer larvae in fields that were next to a field of volunteers. Now, to get the benefit, what you need to do is leave those volunteers in the ground until at least late September. Probably the later the better. But if you remove the volunteers earlier, you may not see an effect, but you may actually also make the problem worse. You also need to leave a large enough area of volunteers, so I would say at least 3 hectares. And by doing this, it protects crops sown from mid-August onwards. It may not provide any benefit for crops sown earlier than that. We've also looked at defoliation to control larvae. So can we remove the leaves in the winter that the larvae are in, and will that reduce pest pressure and yield impact? Well, we looked at this in 2 plot trials in the project, and the data on the right illustrates the results from one of those trials. So this is the larvae per plant at the end of March in the different treatments. So we had an unmown plot and then defoliations in December, January and March. And what we found was that there were significant reductions in larvae where we defoliated, and those reductions increased the later that we defoliated. The results were very similar in the other plot trials, so that in both trials we had a maximum of 55% reductions in larvae. We also saw reductions in stem canker, so up to 50% reductions in stem canker. And that what's happening there is you're essentially breaking that connection between the foliar infection in the leaf in the autumn and the stem. We then took this to farm scale work. So this is an innovative farmer field lab that we ran with on 12 fields where growers either used sheep or topped to do the defoliation. And again, we saw reliable reductions in larvae where they did this. So a mean of 51% reductions where sheep grazed the crop, and a mean of 25% reduction where they topped the crop. Essentially, what's happening here is the larvae are dying either directly through the action of the topper or by being eaten by sheep, but also because they're being exposed to cold conditions and natural enemies. The really major caveat to this defoliation approach is our effect on yield was pretty contrasting. So in the first plot trial that we ran, we saw no significant impact on yield. We did see a slight increase in yield in the earlier defoliations, but in the second plot trial that we ran, we did see significant reductions in yield where we defoliated, and that data is shown on the right here. And you can see that where we defoliated in February, we had a significant reduction in yield, and also where we defoliated in December and removed the debris, we also saw a significant reduction in yield. What we think happened there is that by removing the debris, we were removing a potential nitrogen source for those plants. In the Innovative Farmers Field Lab, we also saw reductions in yield in 10 out of the 12 fields. So what was happening here? Why did in one year did we see no effect on yield, and the other year did we see reductions in yield? What we think was likely to be behind this was the weather. So in the year where we saw significant reductions in yield, we had a very dry spring and a cool start to May, and that would have hindered the ability of those defoliated crops to recover from the defoliation. This isn't the first time people have looked at defoliating oilseed rape, so this has been looked at several times in the last 30 years. It's the first time we've looked at it for controlling flea beetles. The previous work has generally found that there is a negligible impact on yield, and actually some of those trials saw a slight increase in yield. Where we think, if you're interested in doing this, where we think there is going to be the greatest benefit is in large early sown crops, and also potentially crops that are at higher seed rates, because those crops are going to be have a higher level of larvae in them anyway. They're also going to be in a better condition to respond to the defoliation, and also where they're at high seed rates, they're going to be probably less able to tolerate the larval feeding. And also defoliate sooner rather than later to give the crop the best chance to recover. So I would certainly, definitely before stem extension, I'd also really go in before the new year. The last thing I'm just going to touch on here is the effect of larvae on yield. So our understanding of the larval impact is pretty much entirely based on trials that occurred at the beginning of the 80s. And what that found was as larval number increased, we had an increasing impact on yield, so that by about 10 larvae per plant, you would have roughly 0.7 of a tonne per hectare yield response decline. We looked at this again in the previous work to try and validate it, see whether we could see similar results. What we found was a very, very variable yield response, and that's illustrated with this data on the right here. Here we have the yield in different crops that have different numbers of larvae per plant. The different colours relate to different trials, and the lines in them are the fitted lines for each of those trials. Where there is a solid line, that's where we saw a significant response. And in this data set here, there was only one trial where we saw a significant reduction in yield with increasing larvae. In the others, we didn't. Overall, we had 10 trials. Of those 10 trials, only 2 of them did we see a significant reduction in yield with increasing larval number. We have also seen trials where plants have on average 80 larvae per plant, but are still yielding at more than 5 tonne a hectare. Clearly, there are crops that are able to tolerate larval feeding. The question is, what is governing this tolerance? Is it a varietal effect? Is it something to do with the crop structure, the stem width, the crop height? We need to understand more about that. So very lastly, I'm just going to touch on the work that we're doing in a new project. It's AHDB funded, but it also has a very large amount of industry funding as part of it. We're looking at a whole range of things, but the things that we're looking specifically at regarding larvae are the impact of temperature on egg and larval development, That's going to help us time treatments and also predict crop risk. We're looking at the effect of larval number, invasion date, and stem width on crop growth and varietal tolerance. All of this will help us better understand the yield impact of larvae. We're looking at the effect of drill date in trials. That's to validate that survey work that I showed you at the beginning, and fresh data from that is showing that yes, late drilled crops do suffer a much lower larval load. And then finally, we're looking at the relationship between larval scars and larval number. And that's going to hopefully allow us to get a better way of monitoring larval pressure that growers and advisors can do. So I've really barreled through that very quickly. I'm very pleased to clarify any of that or ask any questions in the question session at the end. Thank you for listening, and also thank you to all of these who've either funded the work or worked with us to deliver it.
Thank you so much, Sacha. Fascinating. There's so many intricacies involved in this, and as ever, it's balancing what looks to work for one thing that has an effect on something else. So we've got some really good questions coming in for you, and we'll address those in the question and answer session in a minute. I've got a few questions to ask as well. So we're going to hand over now to Sam Cook, and Sacha will be back in a minute. Sam is a senior research scientist at Rothamsted Research and leads the Eco-IPM group that's looking at an ecological approach for integrated pest management. Sam's focus specifically is on oilseed rape cropping systems and has a real interest in pollen beetle, cabbage stem flea beetle, and managing those pests in the wider landscape. So we're going to hand over to Sam, who's going to talk us through a lot of their research work in the next few minutes. Thank you, Sam.
Thanks. Good morning. Okay, can you see my screen? Hello, can you see my screen?
We can, Sam. If you just want to share, put it on slideshow, that would be great.
Okay, it should be. Sorry.
So unfortunately, I can't actually see Sam's screen. I've got your slideshow up through Heather, and I've got it up through Teresa. I think Heather's going to share it for me, actually. If you just say... I'm
having a fight with the technical. Can you see it?
And just say next slide, Sam, as you go through.
Right, I'm going to step out and off you go.
Okay, sorry, I'm getting two people talking to me at once. Right, okay. I hope you can see can you see my screen? Because I can't. Right, so I'm going to start off by talking to you about why larvae really are important pests in oilseed rape. A lot of farmers are worried about getting their crops eaten, but once you get past that, yes, you really do need to worry about larvae. And a student at Rothamsted and Reading University, Duncan, did a really nice experiment to show that larvae are important. So he did a 2-year experiment. The first year, he tested the response of oilseed rape to various levels of leaf injury, which he simulated with a hole punch. And then in the second year, combined various levels of leaf injury with controlled larval infection. And he put either 1, 5 or 25 larvae in the plant, took them all the way through to yield, and then measured the yield variations at the end of the experiment. Can we have the next slide, please? Okay, Duncan found that high leaf injury, over 90% removal, did not impact the productivity of the oilseed rape plant at the end at yield. So just demonstrates the amazing capacity of the plant to compensate for damage. But he did see responses in yield when 25 larvae, but not 5, were introduced into the plant. The plants were shorter, they produced less flowers and pods, and those pods had lower oil content than the other treatments. So his work suggests that, yes, we do need to worry about larvae. Possibly the 5 larvae per plant threshold is too low, but it is somewhere between 5 and 25 larvae per plant, and they are economically damaging. So next slide, please. Okay, so we are starting to look at what we can do about larvae. How can we reduce these populations in our crops? And we're looking at plant diversification methods or companion cropping, which can reduce these. Things like intercropping, where you have two crops at the same time and they reduce the numbers of pests inside those crops. Here you can see an oilseed rape and pea mixture that our colleagues in Germany are testing. In the middle there, you can see trap crops, which is a very attractive plant that's planted around the outside of your oilseed rape crop. And they can really reduce the numbers. And at the end there, you can see intercropping or undersowing companion crop mixtures. And these methods can improve pest control, pollination, weed control, and provide habitat for increased farmland biodiversity in other crops. But what about oilseed rape? Next slide, please. Okay, so way back in 2005, I had a student again that tested replicated plots of oilseed rape with and without these borders of turnip rape. And these borders were either sprayed or unsprayed with pyrethroid insecticide. Next slide, please. Okay, you can see very clearly that cabbage stem flea beetle really did prefer the turnip rape over the oilseed rape for oviposition and larval development. And I scribbled on these slides, maybe had a premonition that things would go wrong. The scribbles show the numbers of larvae in the borders, the oilseed rape or the turnip rape trap crop borders, and you can see that the numbers in the untreated turnip rape are way bigger than in the oilseed rape crops in the border. Next slide, please. And then you can see that in the oilseed rape plants, all of those larvae are less than in the turnip rape trap crops. So, next slide, please. If we compare just the oilseed rape crops in the middle, we can see that crops with the turnip rape trap crops, that's the two at the end, were significantly lower than the oilseed rape without a turnip rape trap crop. So turnip rape trap crops can reduce the numbers of larvae in the middle of the plants by and reduce them below economic threshold levels. Next slide, please. Okay, and we can see from this experiment, actually, we described Tersilochus macrogaster, which is the main larval parasitoid of cabbage stem flea beetle. We described this for the first time in the UK, which was hugely exciting. And since then, we've shown that actually this parasitoid can have quite an impact on larval numbers. And I'll talk about that a little bit later. Next slide, please. Okay, so forward wind back to Duncan in 2015, decided to try this again. So he tried replicated plot trials of oilseed rape with and without the trap crop borders. And as you can see on the left-hand side, the plant density in the plots with the trap crops was significantly higher than the control plots. If you look really carefully, you can see some green in the middle of the turnip rape trap crop, but there's no green on the right-hand side there. But he also showed that there was a significant reduction in adult flea beetle feeding and larval infestation in those oilseed rape plots with the turnip rape. So it supported our earlier work. However, this was the year just after neonic revocation. We had massive flea beetle populations and they basically ate the whole thing and abandoned the trial in 2016. Next slide, please. But Duncan had another go and did a very complicated experiment, which I won't go into now, haven't got time. But basically, he tested different scenarios of cabbage stem flea beetle control. In what we had in the past was neonicotinoids and pyrethroids. What we had at the time of the experiments, which was pyrethroids and possibly increasing the seed rate. And what we might have in the future, so cultural controls, if we don't have neonicotinoids or any other insecticides, we've got things like trap cropping and undersowing with brassica mixtures, mowing to simulate the sheep grazing, as we've heard from Sacha, and a future synthetic insecticide scenario, which was lumiposa seed treatment and neonic sprays. And you'll see the ones in red, highlighted in red, were the strategies that Duncan found really did reduce the larval numbers in the plants, although they didn't necessarily all have an effect on yield. Next slide, please. Okay, so we're taking forward this idea of undersowing in our new EU-funded project called Ecostack. And a couple of years ago, we had replicated plot trials with several of the mixtures that farmers are using quite widely now in the UK, just to see in very controlled conditions how they're comparing against each other. So we had 6 treatments. We had oilseed rape with white mustard. We intercropped wheat with oilseed rape. We used Berseem clover, obviously had a control, had oilseed rape with a turnip rape trap crop, and we had oilseed rape with wheat broadcasted to kind of simulate wheat volunteers, or this idea that some farmers were finding with straw that fewer numbers of larvae were present in plants that had straw or wheat next to them. And you can see that this actually did pan out. In the top there, you can see we found significant adult feeding with the wheat and the white mustard, and there were significantly fewer larvae in plants that had these wheat plants broadcasted next to them. And if you can have the next slide, please. And this graph here shows the number of larvae that were in the plants. The oilseed rape and the turnip rape plants when they were together in that treatment, and there was no significant difference between the numbers of larvae in oilseed rape or turnip rape, although there were more larvae in turnip rape. And when we can see the oilseed rape and the white mustard plants, there was quite a few in oilseed rape, but way fewer in the white mustard, which indicates that flea beetles really don't like white mustard. So it's giving us a clue as to what the mechanisms of the success of these treatments are. Okay, next slide, please. So our current work is to find out what's going on. So we've got this research evidence to suggest that turnip rape trap cropping, undersowing with brassica mixtures or white mustard, sometimes Berseem clovers, are having an effect on the larval numbers. Also, this undersowing wheat reduces the larval populations, but it's not always consistent. So we're going to need to replicate it a few more times to try to understand why we're seeing these differences and what are the mechanisms of action here. So this year we've been repeating the companion cropping experiments, and we're really focusing now on finding out the effect of the plant density of this. Has the plant density, if you're adding stuff to it, is it just a numbers game? And we're also interested in what's going on with this wheat thing. Is it all cereals? So we've got an experiment that's looking at all sorts of different cereal undersowings. Next slide, please. I mentioned earlier the importance of natural enemies. So we're really focusing on all our experiments, looking what's going on with these treatments and these ways of managing the crop with the predators. So we're really focusing on the role of predators in pest regulation of the adults and the larvae and the eggs in the crop. We're using camera trapping methods to do that with our EU Ecostack partners in Denmark. Next slide, please. And I've got a PhD student, Teresa Ramos, who's looking at the role of parasitism in pest regulation of cabbage stem flea beetle. And she's got a really neat experiment at the moment looking at the effect of crop and landscape management on the parasitism levels of the larvae. And some of you may have been very kind enough to send her samples for this, so we're really grateful for that, for participation. She's looking through those samples and she's looking at the parasitism rates and the distribution of larval parasitism all across the UK. It's a massive undertaking. She's doing this via different methods and looking at DNA metabarcoding with our partners at Newcastle University to help us to find out how this larval parasitism and the abundance of the parasitoid is different across the UK. The next slide, please. And lastly, how you can help. So we do need you to help us to find the answers to these questions and what's going on. We can only get so far with small plot trials, although they're very controlled and we can very, very carefully and robustly compare one treatment with another. They actually are unrealistic. Obviously, we need to scale up to a full field scale in order to really understand that we're not suffering from neighbour effects. So we need to scale up to large field scales. So we need you to help us to do this. If you're interested in running a companion crop or trap cropping experiment on your farm, then please do get in touch if you're willing to host a trial. And next slide, please. And secondly, we're trying to understand the landscape and the effect of that on the natural enemies in the crop and how they're affecting flea beetle control. So we're looking at how field boundary features such as flower-rich margins, hedgerows or tree lines, and the natural enemies they support can mitigate the yield decline that we all know happens at the crop edge. So again, here we really need you. If you've got GPS-enabled combine yield monitor data, please do consider sharing your data with us to help us to understand how this variation and yield can change across the field in relation to the boundary features and the organisms that they support. And I think my last slide is to say thank you. Thank you for listening and bearing with me with my technical difficulties. Loads of people at Rothamsted and our partners at NIAB, Reading, Ferrer and Newcastle University to thank. Thanks a lot.
Well done, Sam. Thank you so much. It's fascinating research that's going on there and really putting, some figures behind some of the kind of conversation that we have about flea beetle. And so it's really good how it's developing through and the research you're doing is practical. And like you say, the more people that can get involved in doing it across the farm scale, the better really we can do. So I've got some more questions coming in for you. So we're just going to go through Charlotte's next slides and then we'll bring you back for our panel session in a minute. So I'm just going to hand over to Charlotte Rowley, and Charlotte is our crop protection scientist at AHDB, leading our research work on pests. And so Charlotte's been behind the commissioning a lot of this research that we're talking about, and just got some exciting other bits that we can't share with you today that would be worth looking at. So over to you, Charlotte.
Thanks, Teresa. Yes, so I'm just going to be giving a really brief update on our current research activity in this area. So, as Sacha mentioned, we've just started a new 3-year project on cabbage stem flea beetle, which is being led by Sacha at ADAS, alongside a whole host of industry and scientific partners. There's a couple of aspects to this. The first sort of work package is all looking around flea beetle biology and getting a better understanding around it. So Sacha highlighted some of the work that we're doing on larval biology, but there's also some work on looking at the factors influencing adult migration. So throughout the migration period, we're going to be trapping adults as they're coming into crops on a daily basis and trying to understand how that relates to weather patterns and whether we can relate that to subsequent damage. So you can see a figure there, which are the results from this year's trapping, and we'll be doing that over the course of the project. Also going to be looking at any differences in adult preference between growth stages to better understand if there is a particular risk factor there. A second part of the work is really putting to test some of these risk mitigation strategies. And so working with all of our industry partners, we're sort of developing a whole network of field trials that are going to evaluate non-chemical control options and really try and build up an evidence base for some of these strategies that we know a lot of farmers might already be trying, and reviewing whether any of these control options can work better in combination together, and whether there's an added benefit there. So again, that's going to be happening over the 3 years of the project. Also, just to highlight our 2 PhDs on flea beetle. At John Innes Centre, we've got Jess, who is looking at the genetic basis of oilseed rape resistance to flea beetle. So she's done some really nice work that has identified some differences in adult feeding preferences between lines from the brassica diversity set at John Innes Centre, and also some significant differences in larval feeding damage. And recently she's been able to take this work out into field trials and assess differences there. So the next step will be to identify the candidate genes involved. Jess is in her 4th year now, so coming to the end of that interesting work, so do look out for the final report on that. The second PhD that we've got at Harper Adams University is Claire, who is looking at novel approaches to flea beetle control. And what she's doing is screening a range of biopesticides against flea beetle adults and larvae. So this includes sort of the biopesticide products and also things like plant extracts. She's had some really good results so far with her initial assays of entomopathogenic nematodes. And you can see a full update of her work at our PhD showcase, which is later today, where she's presenting some data from that. So this project is also, I should say, supported by the Agri-Food Charities Partnership and CSFB. So thank you, that was a really brief update, but I will hand back to Teresa now for some question and answer session.
Thank you very much, Charlotte. And yeah, so much interesting work, a lot of work that's AHDB funded going into cabbage stem flea beetle, and a lot of the projects going. Just clarify again the timelines on that and when people will be reporting on those, Charlotte.
So as Sacha mentioned, we've just finished the 3-year IPM project, so the results from that are all online, and we're starting the next phase now. So this is the first year of that 3-year project. Jess at John Innes Centre is just writing up in her 4th year, and Claire is just going into her 2nd year. So she's got some early results that she'll be taking forward over the next 2 or 3 years.
Brilliant, thank you. A lot going up. Good. So just a reminder, if I can have everybody back on screen for the questions we've got, and just a reminder, if you just click on the poll link at the bottom, we'll share the poll responses in a minute as well. So some questions for you. Sacha, we'll start with one for you. Is there a possibility that there's less cabbage stem flea beetle adult pressure this year, since they destroyed their food supply last year and thus starved and population numbers were reduced? So do you think we've seen less this year because of the effect of last year?
It's possible that we're seeing lower populations because of that. I think a big, I'm not so sure that's the whole picture. I think one of the big reasons we've seen less pressure this year is because we had moisture at the right time during crop establishment, and as Sam's work shows, crops are really able to tolerate a huge amount of feeding damage before the crop is really nobbled. And so I think crops really had the weather there and the moisture that allow them to grow away from it. And also there's a huge amount of very early sown crops, so that by the time the adults came in, they were much bigger anyway.
Yeah. Yeah, it's distinguishing these differences, isn't it, and learning, what one season might have effect on the next, or whether it's an in-season effect. Brilliant, thank you. Similarly related to that one on in-season effects, Sam, there's a question about when Duncan Coston was doing his trials, would he have imposed any drought stress on the plants during the pod formation and fill stages? Because that is gentleman's thinking, well, the interaction between the pest damage affecting the final yield.
Absolutely, yeah. No, he didn't, because the experiment was complicated enough without the addition of another treatment. But that's a really good point. No, he didn't, because it was just simply impossible. But all those treatments were under the same conditions, so we can compare those conditions. We can't answer the question, how does drought affect it when you've got this or that, because that wasn't the aim of the experiment, but that certainly needs to be done. Particularly, I think at the early stages of growth, as Sacha said, the availability of moisture really helps the crop to establish. And I think if you've got a dry conditions plus high flea beetle damage, then that's a recipe for disaster. So I think it would be really good to start to do some drought or some watering experiments at that stage as well. So yeah, moisture conditions, huge effects, but we don't know.
Plenty more research as always to be done on all these things. So similarly related to that, is there any difference, and you're saying about the establishment of moisture being key, so a question about, is there any difference relating to yield with the companion cropping, as that's directly kind of competing with the oilseed rape during those critical stages? Have you seen any yield effects from that?
There's been a lot of concern that planting an undersowing plant will compete for moisture and resources, but we haven't seen that. We've seen that they do reduce the numbers of larvae, but then at the end of the day, there's actually no difference in the yield that we've seen so far on the crops that we've taken to yield. Even though we've reduced the numbers of larvae per plant, we haven't seen a difference in yield. But there has, reduced the numbers of larvae or reduced the numbers of flea beetle adult feeding damage.
Lovely. Thank you.
I was just going to add to that, that in the current project, that's one of the things we're looking at. We've got some farm scale or field scale trials. So these are tramline trials where host farmers are drilling different companion crops in different tram lines, so we're going to be able to get a comparison of that and hopefully get a better understanding of the yield benefit that a companion crop can provide.
That'd be really useful.
The larval, it's not just effect of the larval numbers. A companion crop could be delivering lots of other things, weed control or moisture control or weed suppression. So it could be a number of things, but no difference in yield so far.
Well, that's positive, because I think, with Sacha's that defoliation, it's such that puts that big question over that kind of method of management, doesn't it? So
I found the same in ours actually. When Duncan did, we mowed ours. We mowed them probably a bit too late, and it had a massive effect on yield, really, really massive. And also it made flowering later.
Yes.
And we got absolutely hammered by pollen beetle. And I think this is something that we need to think about as well. Okay, we might be able to get it past the flea beetle damage, but next, are you laying yourself up to pollen beetle damage, which you might do if you're delaying flowering, but also if you're increasing the seed rate, then again, that's going to increase your pollen beetle pressure. So it's a real trade-off and lots of things to think about.
Definitely. So I've got lots of questions coming, so I'm going to fire a few more at you. This is one probably for all three of you. Regarding flea beetle, have you seen any advantages in plant survival or larvae control from applying insecticides? Sam, have you done any work looking at that with insecticides?
Yeah, I mean, pyrethroids generally do reduce the numbers of larvae, but we've never found a yield effect of using pyrethroids. So they don't actually increase the yield in all of our experiments that we've done. Yeah.
And is that because of resistance, or is that because of?
Well, we found that again they do have an effect, a slight effect in reducing feeding damage or reducing larval damage, but again, there's no yield effect. So it does beg the question why they're used in some cases.
So when you've looked at insecticide control for larvae, was that recently, or was it sort of when resistance was less widespread?
It's both. So in 2005 one, that obviously had a point to show that pyrethroids really worked. I mean, there was a massive reduction in the larval numbers in those. It's definitely got worse. That reduction is less. So it does work to reduce the numbers of larvae in the plant. It's working less well now than it did do in 2005. But in neither case did we get a yield response to the application of pyrethroid.
I think the sort of I would agree with that pretty much wholeheartedly. The challenge with pyrethroids is targeting them, because the larvae are difficult to hit. They spend most of the time in the plant. So traditionally we try and hit them at egg hatch, or around egg hatch as best we can. But how do we time that? And especially in recent years where we're seeing egg hatch continuing throughout the winter, whereas traditionally the autumn egg hatch is the main egg hatch, and that's pretty much it. But now, because we have these mild winters, it carries on. So when do you apply it? And resistance, they do have, I think they do carry that resistance that their parents carry. I think they may be slightly less resistant than the adults, but it still is reducing the effectiveness of those. And so, yeah, I mean, I think a challenge is just trying to work out when is best to target them, both for pyrethroids and for any new products that come onto market.
Yeah, as always, more research, and it's managing that, you know, what you're doing, cultural controls, and then what you might need to do, you know, insecticides, or can the cultural controls that you're using, you know, mitigate that need for insecticides, you know, and the balance between the two sometimes as well, isn't it? So yeah, brilliant. A final question for you in this segment, Charlotte, and then I'll pick up on the other ones in our main discussion session, because you've got some other good ones, Sam and Sacha, coming. Is there any collaboration with European oilseed rape researchers? And if not, is there any scope for this in the future? Charlotte?
We don't have anything ongoing at the moment, and Sam highlighted her project, which I believe is EU. But definitely, yeah, we're always looking for opportunities like that where we can leverage additional funding from project partners. So that would be a really interesting area to look into.
Yeah, and certainly like that collaboration across the industry in this has got greater and greater, hasn't it, Sam and Sacha and Charlotte, over the years. So yeah, a lot of coming together for that research, both in the UK and abroad. Brilliant. I'll keep these other questions for when we come back, because there's some of them linked well to Sarah's next one about yield as well. So thank you all very much, and we'll see you in a bit. And we'll now hand over to Sarah Kendall from ADAS. Sarah is a crop physiologist at ADAS and has been working very closely running the Yield Enhancement Network, and specifically on the oilseed rape yen over the last few years. So we're going to hand over to Sarah to talk us through the outcomes of the analysis that's taken place. Over to you, Sarah.
Thanks a lot, Teresa. Hopefully everyone can see my slides. Yeah, so I'd just like to start by thanking Teresa for inviting me to speak in this growing oilseed rape session. It's timely. We had our Yen Awards last Wednesday, of which Teresa hosted. So it's really great to be able to take this opportunity to share with you all some of the lessons that have been coming out of our analysis of the data that's been collected through oilseed yen. Teresa introduced at the start that obviously this season has been quite different for many growers growing oilseed rape, and there are lots of crops around that look forward and potentially are full of potential. And what I hope is that the messages that are coming out of our analysis are useful for you all, whether you have particularly forward crops this season, or perhaps you drilled later and you've got a crop that's looking a bit more backwards. So I just wanted to start by giving a brief introduction to the yen for those of you who might not be familiar with it. So we started the Yield Enhancement Network in 2012, and it was set up really to try and identify and support arable innovators. So it's really been trying to emphasize and promote innovation. So the yen is really an industry effort, and we're really grateful to all the sponsoring organisations for their continued support. Oilseed yen was set up in 2016, so we now actually have 4 years worth of data. And I just really want to try and describe these 4 main pillars of which the yen concept is formed around. So the first thing that's really crucial is that yen is all about science. It's about concepts and understanding. And a lot of what we do in terms of thinking about yields and how yields are formed in the yen is really coming from our underlying physiological understanding that can be found in the AHDB growth guides. The next really important element is this idea of metrics. So it's been really important for us to identify what are the important things to measure, what data do we need to collect, and what are our targets for helping growers and agronomists and the industry really to understand how we can improve yield. And these all feed through into the competition and benchmarking elements of the yen. I've already mentioned that we set up the yen to try and identify who are the innovators in our industry. And this has all been around trying to think about how can we improve yields further. So sharing and developing ideas through networking has been a really important component. And then finally, we want to be able to offer people a way of testing these ideas. So we can generate lots of ideas about how can we improve yields, but it then comes down to having robust methodologies for testing these ideas on farm. And this comes through our farm innovation group way of working, and also other examples in the industry, such as the Innovative Farmers Field Labs as well. So I'm going to now spend some time really summarising the results that have come out of analysing the data so far in oilseed yen. Unfortunately, I wasn't quite quick enough to turn around including Harvest 2020 data in this analysis. So my presentation is going to focus on 3 harvest years, so 2017, 2018 and 2019, of which we have a total of 151 yields. These are from 92 different farms and include some crops in Denmark. And the average yield is 4.8 tonnes per hectare across all these yields. So I think the first thing to obviously point out is that this is a higher average yield than we would expect if we were looking at a data set for all oilseed rape grown across the UK. So I think it's important that that's considered as we're going through. So the analysis that we've done is a multivariate data analysis. And what's really important to clarify at the start of this is that this analysis gives associations. It doesn't show effects. And so what we're talking about are associations of different factors with high or low yields, but we're not suggesting that these factors are the cause of the high or low yields. So we're trying to create a picture really of what factors could be important and how then do we go on to formulate questions and test these in the future. So I just wanted to take a moment really to summarise the top 10 yielding crops from our data set. You can see here that the yields range from 6.8 tonnes per hectare, and then the 10th yield, it had 6 tonnes per hectare. I think what's important to note is that these high yielding crops have been achieved in various locations. So from Kent up to Northumberland and west to Wiltshire. And they're not necessarily all from areas which don't have cabbage stem flea beetle problems. Another thing to note is that there's also a range of drilling dates. So these crops range from being drilled in mid-August right through to mid-September. So obviously high yields can be achieved even in later drilling date situations. And it's also interesting to note that many of these crops were following barley, previous crops. So just to start, I wanted to just introduce the concept of potential yield, which is a really important component of the yen. So what we're doing, and what you can see here in this image, is that we're trying to estimate a potential yield across the UK, and this is based on the amount of light and the amount of water that's available to the crop at each site. You can see from this that these biophysical yield potentials are large, so they average around 11 tonnes per hectare for this medium soil type. You might be thinking, listening to this, 11 tonnes per hectare is so far off what I'm achieving at the moment. But it's to note that we're not suggesting that growers can go out and grow these types of yields tomorrow. This is what could be achieved if we had improvements in agronomy and husbandry and genetic improvements as well. So it's really trying to say, with the weather that I experience on my farm, what is that able to achieve? So although you can see in this figure that potentials are larger in the south and particularly in the southwest, actual yields tend to be higher in the north. And what we see is that the yields that are achieved, that these tend to be from 18 to 81% of the estimated potential yield. So there's a large gap between potential and actual yields, and this indicates that there's large scope for making improvements. I take that with the fact that it's very challenging to grow rape at the moment, and flea beetle obviously poses a significant issue, and trying to close that yield gap whilst dealing with such a pest is obviously really challenging. But what we can say is that high yields are possible in most regions, and it's not just that we see all these high yields kind of congregating in one particular area. So there's obviously a lot of discussion about the impact of cabbage stem flea beetle on yields, but we've also experienced over the last couple of years some challenging weather conditions that have had a big impact on oilseed rape yields. And I think it's really important that we try and think about weather patterns that are conducive or not so for yields. So the most consistent weather associations that we see are the spring and summer temperatures and also solar radiation. So what we found in this analysis is that cool, bright Mays are often associated with higher yields. And this is where we're seeing cooler conditions promoting a longer duration for the crop to set more pods and more seeds. We also see cool June and July and a bright June associated with high yields. And again, it seems that the cooler conditions are useful for promoting canopy longevity, and the bright conditions obviously improve photosynthesis during seed filling. We also see a negative association between December temperature with yield, and this is actually matching up with some of the research done by Steve Penfield at the John Innes Centre. And the other thing that we've seen is that rainfall in February also seems to be positively associated with yield. If we move on now to think about soil characteristics that are important. So the entries in the oilseed yen that we have are mostly on medium or deep clay soils. So we haven't seen a significant association of the RB209 soil type on yield. Soil water availability is really important for yield, and we've seen this positive association with summer water availability. So that's thinking about how much water is held in the soil and also the rainfall. In terms of soil pH, we've seen quite a large range in pH from the yen entrance. So the range is from 5.5 to 8.5. And what's become apparent when we've investigated the data is that actually we don't seem to be able to identify a negative association at these lower pHs, which is interesting and probably something that needs further investigation. In terms of thinking about the nutrients in the soil, we see a positive association with the soil phosphorus index and also a positive trend with magnesium. Again, we see that most of the yen entrants have good levels of organic matter in their soils, and therefore we're not seeing soil organic matter as something that's coming out as being significantly associated with yield. But I think what we have to remember is that we don't always have a big enough range or the stretch to really be able to show some of these factors that we know are important as being associated with yields. We also see positive associations of yield with carbon dioxide respiration as a measure of soil microbial activity. So when we think about varieties, so within the data set, we see about 37 different varieties. The most popular were Campus, Elise and Elga. What we're not able to pull out is a significant association of the specific variety, so whether or not it's a hybrid with yield. This doesn't mean that variety isn't important. I think in our analysis, we see site, weather and husbandry factors as having more influence on yield than variety choice. But actually, it's really key to remember that matching variety traits with the growing environment is really important. And I think what this represents is that many of the growers in Yen are putting a lot of care and thought into their variety choice and choosing varieties that are suitable for their situation. Autumn decisions. Obviously, establishing rape can be a real challenge, and there's a lot of thought put into this part of the period for growing rape. What we've seen is that yields are negatively associated with seed rate. So we lose 0.1 tonnes per hectare of yield per 10 seeds per metre square. It doesn't mean, though, that yields can't be achieved with high seed rates, and we have seen evidence of this in the data set as well. There's a trend for use of cover crops previous to establishing the rape to be negatively associated with yield. And I think this has been highlighted recently. I know this was discussed during the Strategic Farm Week. Maybe there's a lot more we need to be learning about how we use cover crops and the impact that they have on the next crop, for example. And lots of other factors that potentially are important and do have a role, but we're not seeing them coming out in this analysis. In terms of crop nutrition and crop protection, so we don't see a significant association with nitrogen rate. This is really important because what we're saying is that the high yields that are achieved in Yen, they're not just about high nitrogen rates being applied. And we don't see anything coming out in terms of the main type of nitrogen product used either. What is important, though, is the frequency of nitrogen application. So we see an increase, increasing nitrogen applications as being positively associated with yield. We haven't seen associations with sulfur, potash, or phosphorus. This isn't to say that these aren't important. It's probably because the growers involved in Yen are getting the rates right. And also, we haven't seen significant associations with biostimulant use or micronutrient use either. We've initiated Yen Nutrition this year to try and learn more about how we can use the nutrients in the seed or the grain as a way of understanding what's gone on in terms of crop nutrition earlier in the season. And we see significant associations with iron, phosphorus, magnesium, and also with the N/S ratio with higher yields. And interestingly, a negative association with sulfur, which we don't understand yet. And this is something that really I think we need more data to be able to start to look into. It's also worth noting that we see a positive association with the number of fungicide applications and fungicide spend as well. So finally, just to really summarise from a physiological point of view, so we know that oilseed rape is a sink limited crop, and therefore high yields relate to a high number of seeds per metre square. But 1,000 seed weight is important. So a crop that's been well set up with a high number of seeds needs to achieve a good 1,000 seed weight to really see the benefit of that. We've also tried to look at some independent plant measures to try and understand some of the other physiological components. And we see that individual plant biomass comes out as being significantly associated. So I think what's important to note here is that what we're saying is that we've got big plants with a high biomass on a per plant basis, setting lots of seeds and filling those well, and that's where we're getting high yields from. So just to sum up really, so we see that the UK yield potential is 2 to 3 times the national average, so there's lots of scope for improving yield, but that's not without its challenges. Oilseed rape is sink limited, so it's really crucial to maximise the number of seeds per metre square. It's the most important yield component, and really it's about trying to optimise canopy size at flowering. We need to focus on maximising seed filling through longer green tissue duration, and especially focusing on keeping the leaves green, and trying to maximise soil water storage. We know that plant spacing is important, so trying to target a plant population of 25 to 40 plants per metre square where conditions allow is really useful. And what we want is large plants with many seeds. Optimising disease control and nutrition should be a really important focus as well. Here we want to minimise foliar disease and think about careful nitrogen timing to build the optimum canopy and maintain it. We've seen from a nutrition point of view that it's important to avoid phosphorus and magnesium deficiency. So use seed analysis with soil and tissue testing to identify limiting nutrients, and try and mitigate against adverse weather by maximising the soil water storage and root depth to avoid summer drought, and choosing varieties that are less sensitive to warm conditions as well. Finally, I just wanted to mention that for oilseed yen going forward, we've got more of a focus on establishment, and we want to try and make sure that oilseed yen remains relevant to all growers across the UK. So we've introduced an establishment beauty contest, and we're increasing the amount of information that we capture around the establishment period. And this is being funded by AHDB, and you can see more information on the website. And finally, just to thank all the sponsors of Yen for their continued support. And I'll finish there. Thank you very much.
Thank you, Sarah. Yeah, a lot to take in as ever, and a lot, you know, that is known, and still some of the questions that aren't known. And like you say, I think that'll only build, won't it, as we go forward. So thank you. And we've got lots of questions coming in for you. So if we bring back the rest of our panelists, we'll get through to those in the time that we've got left. Just before we go into the questions, if we could bring up the results to the poll, it'd be quite interesting just to see what what our audience are finding in terms of larval numbers as well. So less than 5 a plant is our dominant one at 43%. But if I could just ask my other panelists to mute their microphones, and just give me a bit of feedback, that would be great. And 27% of you haven't looked yet. Some that haven't got any larvae, and some that have got, so quite a mixed number of you across the page, but there is definitely larvae out there, and some of you that will go and have a look maybe and think about what we're doing after today. Thank you. Great. So if we can have us back as a panel session. So we've got a variety of different questions coming in, which we'll go through in a different way. So I think one for you first, I might start with you, Sam, for this one, but Sacha might have a comment on it as well. Is there a difference in attractiveness of oilseed rape plants to flea beetle depending on their growth stage? Somebody here has been seeing adult beetles attack plants at cotyledon stage, but then leaving plants at the 2 true leaf untouched in the same field. Is that something that either of you have looked at there?
Yeah, we've had a little look. Duncan had a look actually in controlled conditions. So in controlled conditions, we actually found that they preferred the older leaves compared to the younger leaves. But yeah, in the field, it seems to vary, and it depends what's out there as well. I know that this person said that the beetles left the field when they're at the 1 to 2 leaf stage. They do seem to switch. They come in later, depending on what else is around them. I found that, but I think that they prefer the older leaves. That's what our research has suggested in controlled conditions. But we've started to look at that in the field. So I don't know what Sacha thinks.
You're interesting. Sorry, Sacha.
Sacha, you're muted.
Yeah. Okay. Yeah. More technical issues. I think we've all I can say is anecdotally, we think that potentially they do prefer the younger leaves. This wasn't controlled, so this was where a grower had one of the growers that we work with. He drilled his field, but one part of his field hadn't established very well, so he then went in and just broadcast some seed into that corner, and this chap he's really engaged. He puts out water traps. He moved over some water traps to that area of the field, and he saw a huge spike in flea beetle numbers in that area of the field, and the crop didn't establish. So that's all we've got to go on. Yeah, we definitely think there's definitely something going on in terms of growth stage preference, whether it's one way or another, and it's one of the things that we're going to be looking at in the new project. So it's a piece of work that's been delivered by us and Harper Adams, and it's one of the bits of the project that Harper are going to be looking at is growth stage preferences, but it will be in controlled conditions, so it will be interesting to see how it tallies with Sam's work.
Yeah, so it sounds like one for us to note down as well, Charlotte and Sam, to take forward, and yeah, it'd be interesting to hear the work of the Harper work. Brilliant. Just related to that, Sarah, has any of the work been done in terms of yours, looking at oilseed rate or the frequency of previous oilseed rape crops in the rotation?
Yeah, so we ask the entrants to state when oilseed rape was last grown in that field. So it's been something that we've been able to analyse within our analysis. It's not something that came out as having a significant association, but I think despite that, we do know that there have been instances where some of the growers who have achieved very high oilseed rape yields in the yen, there has been a really large gap since the last oilseed rape crop was grown. And I think, you know, some of these things we can't show from our analysis that they're important, but it definitely doesn't mean that because we haven't shown it and I haven't discussed it here, that something that would make kind of legitimate sense isn't important. So, yeah.
Brilliant. Something that doesn't make legitimate sense, I wish we could control, but we've got a couple of questions here based on the weather and predicting the weather. Kind of a comment on yours, Sarah. You know, since we don't know anything about the weather, how do the associations with weather help us optimise the yields? I'll ask you to do that one. And related to that, Sam and Sacha, just so you can think about it, have we got anything, and Charlotte, chip in here about predicting flea beetle populations based on the weather? So I'll let Sarah, if you want to answer the yen associations one first.
Yeah, so it's great to be able to look back over a year and talk about all the things that explain yields from a weather point of view. And obviously really difficult from a growing rate perspective because you have no idea what weather is still to come that season, and it does have such a big effect. I think though what we can learn from the analysis and from those kind of components of the weather that are important is actually how can we help crops to mitigate against the negative impacts of some of these you know, weather experiences. So take the waterlogging that many rape crops experienced last year. Obviously, you know, we're trying to think about how do we maximise rooting and get roots down quickly so that wet weather over winter doesn't have a big negative impact on this. It's thinking about soil as well. So, you know, if we know that wet winters are not helpful in terms of yields and we need to be doing things to to avoid where possible. Obviously, getting drainage right, getting soil structure right is really important, and linking that really to our understanding of water availability. And so if we're seeing drought conditions in the spring or, during seed filling, actually making sure that the crop has adequate roots to capture as much water as possible from the soil is really important.
Great, thank you. Charlotte, is there anything we know yet about predicting flea beetle populations based on the weather or work that's going on in that area?
It's quite hard to do. I mean, we are limited in terms of how far ahead we can forecast accurately the weather. I mean, it would be amazing to be able to drill according to when we predict flea beetle will migrate, but it might not be a realistic goal. What we might be able to do is rather use the weather as a way to anticipate the flea beetle activity in the field and anticipate future stages. So, for example, are they having a prolonged egg laying period, and when do we expect the larvae to be moving into the crops? And that approach might be more of a useful area for our work. Sacha, you might want to jump in about the work we're doing on the adult monitoring this year.
Yeah, thanks, Charlotte. So, the big question is one of the big questions is can we predict adult migration, and it's one of the things we're looking at. So we are monitoring this year around a dozen sites. We'll be doing the same thing next year and the year after, and many of those sites will also be monitored every day, and seeing whether we can relate any of that the activity in terms of flea beetles with specific weather events, so dry periods, peaks in temperature in particular days, or is it an average temperature across a day? And ultimately, that will help us understand where we need to apply some IPM methods, and also, as Charlotte said, we can also use it to help predict other things, so the levels of damage in the crop potentially, and also the levels of larval pressure going on into the autumn.
Okay, thank you. I think, like we've said a lot, Charlotte, haven't we? Like the understanding of that biology and the life cycle and how that's working in the field is something we want to do a lot more work on. I know, Sam, you're doing a lot of that as well. So brilliant. I've got a couple of...
There is a commercial product out there called Zavio, and I've done some work with them in the past, and they do quite accurately predict the migration activity and development of the eggs and larvae. So there are commercial tools out there, but not for the UK, unfortunately. So we've got some work to do to make our own, probably.
Brilliant. Just a couple of specific questions from your presentation, Sam, if I can. The turnip rape trap crop that you've used, has that been done in a large-scale field trial? And if so, was the effect seen, and how large was that border? And would a wider boundary of the turnip rape improve the effect?
So yes, for pollen beetle. So we've done it on a full field scale for pollen beetle. We're working on 10% of the crop area for pollen beetle. So it was a 6 metre border in a 1 hectare, a very small 1 hectare size field. Pollen beetle moves differently to flea beetle though. Pollen beetles colonise the field from the crop edge. We're less sure that happens actually with flea beetles, and we seem to think that they come more towards the centre of the crop. So actually putting them at the borders might not be necessarily the best idea, and maybe that's why strategies like undersowing the brassica mixtures tends to help slightly. So obviously the bigger it is, the more they're going to soak up, and the effectiveness depends on the size of the population. So it's quite a complicated answer. But yeah, if you've got a small population, you could get away with a small trap crop. But if you've got a big population, you'll need a bigger trap crop to reduce the numbers. So there's a lot of work to be done to optimise the strategy.
Yeah, and that difference, like you say, between trap cropping versus intercropping, you know, or companion cropping to do. Just a quick question, a quick answer on why would that wheat undersowing have an effect on the larval numbers?
Yeah, don't know. So I just want to go back and say thank you to, we have got one farmer that's trialling trap cropping this year, turnip rope trap cropping. So thank you. We do need some more to get the replicates up. We don't know, and that's one of the main you know, points in my slides, that now we're really looking towards the mechanisms. So it could be it's confusing it could be it's repellent, it could be things that, you know, leachates in the soil, or we don't know. Is it just wheat? So we're starting with that one right now. It doesn't seem to be, but yeah, we're working on it and we'll let you know.
No, it'd be great, because it's these kind of practical questions. We've got a question in here about costs, you know, and the cost for it, and actually if we can leave volunteers, you know, and that has the same effect, and that will reduce the costs. And, you know, to reply to the person that's asked about costs, it's something that we're looking at. Sam has said that through the Ecostack work with they're doing in Germany, they're looking at the costs, and I think it's something, Charlotte, that we'll pick up on more, you know, as and when we've got a series of options that we know work, we can cost them up and look at the cost benefits. So just a final question, Sarah, saying there must be a seed rate curve for yen or seed rate crops. I know you've kind of found that the larval numbers and, you know, the effect of seed rate. Do you want to just answer that one quickly, and then we're nearly there?
Yeah, so in my slides, I think I just showed a linear response for the seed rate data from yen. So it probably would be possible to fit a curve to that. But I think the message is that definitely we're seeing a negative association with increasing seed rate. And the thing that's nice about this is actually, you know, the data that Sacha presented earlier shows that from a kind of managing the larvae point of view, actually having high plant populations isn't beneficial. So although it's a challenge, isn't it, to establish rate at a low plant population where you know that you may lose plants due to adult feeding damage, actually it's all quite consistent in terms of our understanding of the physiology and the fact that large plants are potentially you know, the ones that will go on to yield more. What we're looking for is big, strong plants. And whether we have indications from a larval perspective that these plants could be more tolerant as well, and may be able to sustain higher larval populations. I don't know whether you want to comment, Sacha, on that.
You're on mute, Sacha.
Yeah, there does seem to be some suggestion that there is something to do with the crop architecture that's making it more tolerant. I think, as I said earlier, one of our field lab sites, they sow at very low seed rate. They have these very large plants by the time spring comes around, and they were able to tolerate very high numbers of larvae with very little impact, if anything, on yield. And so yeah, I would agree with Sarah there, and it's just about trying to work out exactly what it is about that crop architecture that is conferring that tolerance.
Brilliant. Thank you all very much. We have got some more specific questions about pigeon control and chalky soils and the relationship of nitrogen fertilizer and what you're finding in the yen and how you measure that. You know, how many flea beetles there are at harvest and the effect, and, you know, whether we can use that and kind of whether you can look at barley volunteers as well, Sam. So we'll take some of those specific questions. We'll ask the speakers, and we'll make sure that the answers to those are available back under here with the recording. So thank you all very much for your questions. Thank you for engaging, and many thanks to our speakers for sharing their findings, sharing where we've got to, and possibly some of the questions we've got yet to ask, Charlotte. So brilliant, thank you. I'm just going to go through, if you'd like to share my slides, just for the last couple of bits for you to take away. Please. So just a couple of things to mention in terms of further information. The oilseed rape growth guide, which is heavily linked to what Sarah was talking about and kind of monitoring that crop through the season, is available as a download at the bottom of the page that you're watching us from. All the oilseed rape disease management guidance is available online. And the cabbage stem flea beetle is both available if you type in those URLs. Equally, I have put a copy of the flea beetle, the latest integrated pest management report that Sacha mentioned at the bottom, as a download as well. If you have any questions, I'm sure our speakers today will be happy to answer them. You can chat to them directly through the Agronomy Week platform. I'm happy to take any forward, and Charlotte Rowley, who's our research manager for pests, and Catherine Harries, who's our research manager for diseases, plus our recommended list team, and others are happy to help from AHDB as well. So please get in touch with us and take the questions forward. We've also launched our arable review, which is detailing all of the AHDB research projects and where your levy funded research is going. That's available to download and as a hard copy if you'd like to email me. Please do open that, have a look. There's an overview of all the research that's gone on. There's a list of all the projects, the reporting deadlines, and they're all in there. So every one of you, please, should open that. It's a very valuable piece of work and give you an insight into the work going on in the industry. So it's just a final thank you from me to you all for joining us for this event. We'd be really pleased to hear how you found this event by filling in our survey at the feedback form or rating it again further down in the session. If you collect basis and Aroso points, the codes that you'll need to go and enter them, if you go into the left-hand panel, you'll see there's a link. B12H is your basis code and N12H is your Aroso code. Again, any questions, drop me or the helpline a question. There is our PhD showcase, mentioning one of the PhDs that Charlotte said about earlier. That's happening at 4 o'clock today, looking at the novel approaches to cabbage stem flea beetle, and that will be available for the rest of the week as well. And please do come back and join us for our next session at 2 o'clock. Lovely to see you all. Thank you so much. And a big thank you to our speakers, to Sacha, to Sam, to Charlotte and to Sarah. Thank you for sharing your research insights, and we look forward to following on for these in the coming year. Thank you, everybody. Goodbye.